This is Aniket Kumar Singh and I'm right over here with your NEET and AIIMS career. So I'll be taking in this video the very subtopic of the chapter 2 of 11th course, namely Kingdom Monera. You know, history of taxonomy and all मुझे पता है NCERT में but I'll be discussing it later on. Okay, so let's directly start with Kingdom Monera. I'll be discussing all the 30 years questions right over here with my theory and giving it to you. Right, so you know, try to just read only those particular concepts जो मैं बता रहा हूँ. Don't go in much detail. Okay, Kingdom Monera especially, there are some books, namely Pradeep, True Man's Biology. Please, mat padhna उन books को. You know, you are here for preparing your NEET and AIIMS, not for your BSc, right? So, just think that I want to clear it to you. Okay, so let's directly start with our Kingdom Monera. Right, so here's your Kingdom Monera. So now basically let's discuss directly the basic classifications. You know, Kingdom Monera के कुछ basic characters and that you have to very first, you know, Monerans are going to be your prokaryotes, right? So basic characteristics you should know is सबसे पहले cell wall. Some very basic characteristics. Okay, cell wall. The cell wall over here for Monerans, अगर मैं Monerans की बात करूँ, the cell wall of Monerans is going to be made up of murine. Okay, it is something which is made up of murine. Very important. Murine is a type of peptidoglycan, very, you know, strictly speaking a carbohydrate, right? So murine is going to be in general, but there are exceptions like archaebacteria and all. Cell wall is made up of murine, peptidoglycan. Cell membrane की अगर मैं बात करूँ, the cell membrane in case of prokaryotes is something which is made up of lipoproteins. In case of eukaryotes, the cell membrane is again made up of lipoproteins. So can I say one thing, that the cell membrane of prokaryotes is going to be analogous to the cell membrane of eukaryotes, right? So ये NCERT example का एक question है, right? So you have to remember this. Cell membrane of prokaryote is going to be analogous to cell membrane of eukaryotes. The very another thing that you should know is going to be the cytoplasm. The cytoplasm has many organelles, but remember one and the very important thing, all the organelles, जो भी तेरे cytoplasm में organelles हैं, they are going to be membrane-less or lack of membrane-bound organelles. Lack of membrane-bound organelles. ये बहुत अच्छे से तुम्हें याद रखना है, ठीक है? The cytoplasm of prokaryotes have no membrane-bound organelles. ठीक है? So the particular kind of nucleus condition in case of prokaryotes, the particular type that is present in cytoplasm is having some names. So you have to remember it's given in NCERT practical as well as NCERT example. Incipient nucleus, genophore. Incipient nucleus, genophore, then incipient nucleus, genophore, then fibrillar nucleus. ये सब some names हैं that you should know as per me. And another thing, और कुछ नहीं होगा, right? इसके बाद you can remember like very famous name nucleoid, okay? एक और चीज बहुत ही importantly you have to remember that the cytoplasm of Monerans is going to have no true chromosomes. बहुत important है बेटा. That's something which is very important. The cytoplasm of Monerans is going to be present with a type of chromosome which we call it as false chromosomes. These false chromosomes are made up of, these false chromosomes are something which is, you know, these false chromosomes are made up of non-histone proteins. Non-histone protein का एक example है, that is called as polyamines. Non-histone protein plus a type of DNA. This DNA is going to be very important. NCERT exemplar DNA. This particular type of DNA is going to be naked, you know, or free floating. Naked, free floating. DS circular, double-stranded circular DNA. The DNA which is found along with the non-histone proteins like polyamines is going to be DS circular, naked or free DNA, right? So it has to be understood by you. This is one thing, okay? Then there is no true chromosome. This particular type of, you know, condition is this is only called nucleoid condition. This is called as nucleoid condition, right? This particular nucleus is having no membrane. This is one thing. Then ribosomes, you can say for bacteria, it's, I just hope you know, 70S ribosome. We will be discussing about S. What is S? Rydberg unit in cell bio chapter. 70S ribosomes. Okay, that's one thing. So उसके बाद जो तुम्हें याद रखने बच्चे वो है कुछ examples as per me. तो देखो यार, actinomycetes, then rickettsia and chlamydia. Spelling? Chlamydia. ये examples बेटा याद रखो. These are some examples of किसके bacteria के. These rickets is going to be a disease of vitamin deficiency. Rickettsia is a bacteria. Okay. Chlamydia is something again a bacteria, not a algae. It's confused करता है. Actinomycetes is not a fungi, is again a bacteria. So you have to remember this particular examples. जो पूछे गए हैं जिसमें बच्चों ने confusion हुआ है. So that's why I'm giving it right away. Okay, so these are the general characteristics. I guess I've discussed major part. Then let's discuss each and every bacteria in a bit detail. सबसे पहले eubacteria, जिसका सबसे important है NCERT ने बहुत कुछ दिया है, बहुत ध्यान से दिया है. eubacteria, you have to remember eubacteria. Eubacteria is going to be something which is normal. You have to remember first कुछ history है eubacteria से related. Leeuwenhoek. There was a person known as Leeuwenhoek. There was another person who was known as F.J. Kohn and very famous person Berkeley. Okay, very famous person Berkeley, right? अगर तुम्हें थोड़ी सी भी hobby है ना books पढ़ने के related with biology, you can just know this person. Trust me. कोई भी book हो bio का, कोई भी book, very good book, उसमें इसका नाम पक्का होगा. Trust me, it's that important. Leeuwenhoek was that person who सबसे पहले discovered the particular type of bacterial culture. Okay, he discovered bacterial culture. He told, उसने बताया कि बैक्टीरियाज करके कुछ होते हैं, कुछ छोटे छोटे ऑर्गेनिज्म हैं that are present. उसने कहाँ दिखाया था? उसने दिखाया था teeth scum में, but he observed it for the first time in rainwater. That is very important and that confuse you a lot, right? तो हमेशा याद रखना, the person who discovered the bacteria was Leeuwenhoek. He observed it in rainwater and then proved their presence in the teeth scum. That is important. F.J. Kohn was that person who coined the term bacteria. Okay, this is something which can be asked because पूछा गया 2013 NEET में, इसलिए coined the term bacteria, right? And another very important Berkeley. There was a very famous book, namely Manual of Determinative Bacteriology. Manual of Determinative Bacteriology. This is that particular book which is now at present time considered to be the Bible of the study of bacteria. Why? Because this book has everything about bacteria. You want to know everything about bacteria is being given in this book. So we just know about this book. So that's the name of the book, Manual of Determinative Bacteriology. It was given by Berkeley, and that's why he's he is called as the co-founder of the studies of bacteria, kind of, right? So then this was a basic history particular type. अगर मैं shape के basis में type करूँ, देखो, the bacteria is basically classified in many types. Okay, on the basis of shape, the bacteria is classified in 4 major categories, right? One is going to be your coccus, another is going to be your bacilli or bacillus, another is going to be spiridium, another, if you want to say, is going to be comma shaped or vibrio. It's going to be important, right? और तुम्हें coccus में एक चीज याद रखनी है, वो कुछ नहीं है इनमें. This is going to be spherical in shape. This is going to be rod shaped. This is going to be spiralum, right? Spiral, curved, shaped. This is going to be comma shaped. So many examples, as for me, I don't need to give you the diagram, but anyway it's given in NCERT. It's going to be this one, kind of, right? And bacillus is very important diagram. Most of you just leave them because it's important. That's why I'm telling. It's even again repeated in class 12 biotechnology chapter, as well as strategies of food enhancement. तो ये तुम्हें डायग्राम बेटा याद रखना है. क्यों बताता हूँ मैं? इसमें एक चीज है जो मार्क करने के लिए AIIMS 2008 ने पूछी थी. That is going to be your spore. It's very important. Remember the particular type of this one, right? The head of the bacillus bacteria is going to be, अगर मार्किंग के लिए पूछे, this is going to be spore. It's very important. It's given in NCERT. Direct diagram I'm giving, right? फिर spiralum, spiral shaped, ये भी important है. क्यों बताता हूँ? If you just see, this is the kind of spiral shape, they are just coiled kind of, right? they are curved or spindle like. हमेशा याद रखना, these are those bacteria that are having something like this. They have some special projection which we call it as flagellum. It's very important बेटा, ये भी मार्क करने का हो सकता है, since ये पूछा है तो ये भी पूछ सकता है. तो spiral bacteria, all spiral bacteria will have flagellum. हमेशा याद रखना. इसका मतलब क्या होगा? Can I say all spiral bacteria are going to be motile in nature? It's again NCERT example. Then comma shape is going to be simple, right? Comma shape. It's not this one कुछ ऐसा. So kind of. अगर मैं stick to NCERT, इसमें कुछ मार्क करने का, तो मैं वही बताया, सबसे important है ये, and ये बहुत important है because इसमें मार्क करने के लिए दिया हुआ. It's going to be very important. Then उसके बाद जो तुम्हें इसमें याद करना है is going to be examples, right? So coccus, Diplococcus pneumoniae. Diplococcus pneumoniae. Griffin's experiment का raw material was this, Diplococcus pneumoniae. उसके बाद अगर मैं bacilli में हूँ, so it is going to be E. coli. It is going to be E. coli or Bacillus anthracis. Bacillus anthracis. Spirulum bacteria. Yeah. So Bacillus anthracis, E. coli, Bacillus anthracis will be the spirulum bacteria. Sorry, bacilli bacteria. Spirulum bacteria, you can say very important example. NCERT example. Treponema. Very, very, very important. Causing night sleepiness, sickness, and there is very simple volutins, spirulum volutins. So yeah, आसान है याद रखना. The very important that is usually asked is going to be your treponema, E. coli. Very, very, very important. Comma or vibrio, vibrio cholerae that causes cholera too. So these are some examples that you should. सबसे important is E. coli and treponema. That is usually asked. Again bacteria on the basis of bacteria की classification है on the basis of the particular. This one, flagella. On the basis of the position or the number of flagella present, they are classified. There is a mnemonic for this. मेरा दोस्त है एक अमल करके, ठीक है? उसके बाद उस पे CP डाल दे, ठीक है? Kind of, co-assistant of a principal, kind of. चमचा तो है ही वो. So अमल, CP. Remember, अमल. A for is going to be atrichus. हमेशा अमल याद रखना. M for monotrichus. Right? A for amphitrichus. L for lophotrichus. Then C for cephalotrichus. And P for peritrichus. It's going to be very, very, very important that you should know over here. It's very important, right? Remember, atrichus. When no flagella is present along the bacteria, that is called as atrichus. Monotrichus, a single flagella is present anywhere around the body. Amphitrichus, when single flagella are present on two sides of the bacteria, that is called amphitrichus. Lophotrichus, when bunch of flagella is present on both the side. When bunch of flagella is present on both the side of the bacteria, that is called as lophotrichus. Cephalotrichus, when bunch of flagella is present along one head, that is called as cephalotrichus. Peritrichus, peri means from all the side, right? From all the side, bunch of flagella is there. From every side there is going to be bunch of flagella. So you have to remember examples, I guess, for this. That is going to be important. For atrichus, remember micrococcus. The example is going to be micrococcus, right? For monotrichus, you have to remember your very famous Vibrio, right? Vibrio cholerae. Then for amphitrichus, you can say one of the examples is going to be your Nitrosomonas. Nitrosomonas. Then for lophotrichus, if you wish, you can remember Salmonella. Salmonella. Cephalotrichus, if you wish, Cornybacterium. The example is going to be Cornybacterium. For peritrichus, E. coli and Salmonella typhi. These are the examples. So I guess you should note this particular thing. So it's going to help you a lot because this is directly from NCERT example and it helps you a lot. Okay. So this is one thing. So these are the basic things for kingdom Monera, that is eubacteria. Now eubacteria में एक common चीज और है. There is the flagella. Very, very, very important. The structure of flagella is going to be very important, right? So just note over here. I'm just discussing the structure of flagella. ठीक है, द स्ट्रक्चर ऑफ फ्लैजेला, दैट इज वेरी इंपॉर्टेंट. अगर मैं फ्लैजेला की बात करूं, देखो ध्यान से, द फ्लैजेला इज ब्रॉडली डिवाइडेड इन थ्री मेजर पार्ट्स, राइट. द फ्लैजेला इज डिवाइडेड इनटू थ्री मेजर पार्ट्स. वन इज कॉल्ड एज द बेसल बॉडी, द अनदर इज कॉल्ड एज द हुक, and the third part is something which is called as the filament, right? So हमेशा याद रखना NCERT exemplar plus AIIMS 2002 का क्वेश्चन है, the particular rod shaped structure present in the flagella of the bacteria is going to be basal body. This is going to be the rod shaped structure, whereas the filament is going to be hollow cylindrical structure. It's NCERT exemplar. This is going to be very important. Basal body is going to be rod shaped. Filament is going to be hollow cylindrical structure. It's really very important. I'm telling directly. It's NCERT exemplar है, पूछ सकते हैं, right? So now let's discuss all the major part in a bit detail. Okay. So basal body is going to be rod shaped structure. It is going to be proteinaceous structure. इसको याद रखने का एक ट्रिक है. देखो यार, मैं एक डायग्राम बना रहा हूँ so that it will be easy for you to remember. Okay. कुछ ऐसे, right? Remember one thing that is very important. अगर there is a bacteria have a cell wall, right? तो सबसे पहले जो बैक्टीरिया को कवर करेगा वो होगा सेल वॉल, एंड उसके बाद आएगा सेल मेम्ब्रेन. If I move from outside to inside, the very first part I will be getting is cell wall, right? And very second part I will be getting is cell membrane. It's very important for you to understand right over here. तो मैंने ये structure बताया, उसके components I will be discussing it. So the answer, the space between cell wall, this is the particular space, the space between cell wall and cell membrane is something which is called as periplasmic space. The space between cell wall and cell membrane is called as periplasmic space. NCERT exemplar, this is going to be analogous to lysosomes. बहुत important है बेटा ये, very important. The periplasmic space in case of bacteria is going to be analogous to lysosomes because this functions are exactly similar to eukaryotic lysosomes. ठीक है, एक और मैंने बताया था, right? It's like that lipoprotein वाला cell membrane is going to be analogous to human cell membrane. ठीक है, तो ये सब याद रखना है. It's NCERT exemplar. Then देखिए, these are some rings. Okay, the cell wall have two types of ring. The first ring is called the L ring, another is called P ring. Cell membrane has two type of ring. One is called S ring, another is called as M ring. Okay, and then ये उसको connect करते हैं दोनों साथ में. Okay, so ये तो मैं याद रखना है. It's going to be very important right over here. So next is उसके ऊपर से एक flagella जाएगी. This is going to be one flagella. Okay. So हमेशा रखना. क्यों बताया ये मैंने? Because I guess AIIMS 2015, or I don't remember exactly, but I remember AIIMS 2017 have asked this question: the gram-positive bacteria. On the basis of gram strain, I will be discussing it. There are two types of bacteria. One is called positive and gram-positive, gram-negative. हमेशा रखना. Gram-positive bacteria is present only with this one, S and M rings. Only with S and M rings. इसका मतलब क्या हुआ कि L and P rings are going to be absent. इसका एक और मतलब क्या हुआ? The cell wall is not going to be completely well developed. I'm not saying कि the gram-positive bacteria में cell wall नहीं है. It's not well developed as normal bacteria, and that's the reason why it retains the stain of gram. That is very important. I'll be discussing it right over there. तो मैं ये structure हमेशा याद रखना है. The rings ये question आया था AIIMS 2017 में. The present is S and M ring. That is going to be very important. L and P rings are also called as outer rings, right? And this is also called as the inner rings. So the inner rings are only present in case of gram-positive bacteria, whereas in case of gram-negative bacteria, both are complete as outer and inner rings. Both are going to be present. Okay. So this is the first part, that is going to be basal body. Hook. The function of hook is to connect the basal body to the filament. Another thing is you have to remember filament. Remember, मैंने बता दिया the structure of filament is going to be hollow cylindrical structure. हमेशा याद रखना, this filament is made up of protein monomers. This filament is going to be made up of protein monomers, and these protein monomers जो है ना तेरा, it is made up of flagellin protein. It's very important for you. The monomer, it's NCERT exemplar again. The flagellin protein, it is made up of flagellin protein, and this flagellin protein is going to be a contractile protein. ये question again आया हुआ है. I don't remember exactly, but I guess it's AIIMS 2012 का question है या PMT 2008 का question है. It's going to be the contractile protein which is being given. The flagellin protein is going to be a contractile protein. I'll tell you why I'm saying this. There is something called as pilli, जिसको सब में doubt होता है, right? सबको इसमें doubt होता है. Pilli and filament. I'll just write it as a comparison ताकि पढ़ने में आसान जाए. Pilli and filament, right? बहुत doubt होता है सबको, so I'm just telling you right over here. Remember, pilli are always numerous, whereas filament is going to be less, 1 or 2 in number. ठीक है? Whereas, अब देखो ध्यान से, these are going to be numerous, these are going to be 1 or 2 in number. Then pilli is made up of very important pillin protein. Filament is going to be made up of flagellin protein. अब इन दोनों में difference क्या है? दोनों protein से बना है, right? Remember the difference. Flagellin protein is going to be a contractile protein, whereas the pillin protein is going to be non-contractile protein. It's very important, right? Flagellin protein is going to be a contractile protein, whereas pillin protein is going to be non-contractile protein. Another thing that you wish to is the pilli. The pilli is out of 2 types that you have to remember right over here. One is called as the longer pilli, another is called as the shorter pilli. Very important, हमेशा याद रखना, the longer pilli is also called as F or sex pilli, okay, because it is used during conjugation, transduction and all. I will be discussing it. Shorter pilli is something which is used for attachment to the host. So can I say one thing? This is that particular pilli that attached to the host. So this is the pilli via which bacteria infects the organism, right? So this is going to be the infective pilli. Okay, this is going to be the infective pilli. The infective pilli is going to be the shorter pilli, and the sex pilli or the F pilli is going to be the longer pilli. So these are the filament. Filaments के कोई types नहीं है. तुम्हें बस यही याद रखना और कुछ नहीं. ठीक है? So I guess and हमेशा के हमेशा याद रखना. Pilli plays no role in motility. They plays no role in motility. But filament, the main function is motility, mainly for motility. ठीक है? Or yeah, there is one thing. This is going to be pilli, filament. There is one more term that confuses you a lot. That is called as fimbriae, right? So fimbriae confuse मत होना. हमेशा याद रखना. Fimbriae is something which is used for colony formation. हमेशा याद रखना. If fimbriae is absent, Kerala PMT 2019, this one, sorry, not Kerala PMT, Kerala this one, KCET 2019, the fimbriae are going to be absent, then what will not occur? There will be no formation of colonies by the bacteria. So this is something which is again very important. So I guess you should try to make this out. It's very important. Okay. So yeah. Okay, so this is one part. Structure of flagella is done. Let's now move on to another topic that is going to be the structure of cell envelope. It's going to be very important. The structure of this bacteria. Okay, देख. अगर मैं outer to inner हूँ, हमेशा रखना. From outside to inside, the very first layer for eubacteria is going to be glycocalyx. The very second layer is going to be cell wall. And the very third layer is going to be cell membrane. It's NCERT liner. हमेशा याद रखना. Glycocalyx, the outermost layer. The innermost layer is going to be cell membrane. In between is going to be cell wall. These three all together make something which we call it as cell envelope. NCERT liner, NCERT exemplar. It's very important. Cell envelope consists of 3 layers: the outermost glycocalyx, the innermost cell membrane, in between is going to be the cell wall. हमेशा रखना the cell envelope. All these layers is something which is these perform distinct function. All these layers have distinct functions, but unite together. These all of them have distinct function, but unite together. Glycocalyx, cell wall, cell membrane unite together to form a cell membrane which functions for protection of bacteria, which functions for protection of bacteria. It's again NCERT liner. Each layer is going to perform distinct function but unite together to form a cell envelope which functions to protect the bacteria, right? So it's important right over here. Then glycocalyx आते हैं, ठीक है, एक एक करके सारे डिस्कस करते हैं. Glycocalyx, अगर मैं glycocalyx की बात करूं बेटा, it's going to be very simple. Just listen to me. The glycocalyx, in the sense, see, glycocalyx have two types of layers, right? So first layer right over here is called as the slime layer, and the very second layer is something which is called as the capsule. हमेशा याद रखोगे, glycocalyx slime layer, slimy, is going to be loose. It's going to be loose and thin. Glycocalyx capsule, capsule is something which is hard. It is going to be tight and tough. It is going to be tight and tough. Glycocalyx slime layer loose and thin. Glycocalyx capsule is going to be tight as well as tough. क्यों होता है? AIIMS assertion and reasoning. Because the slime layer, if I'm talking about the slime layer is something which is made up of thick. हमेशा याद रखना, ग्लाइकोकैलिक्स जो है एक कॉमन चीज होती है, दैट इज गोइंग टू बी पेप्टिडोग्लाइकन. ठीक है, दोनों के दोनों पेप्टिडोग्लाइकन से पक्का बने रहेंगे. बट along with that, capsule is more tough, right? देखो उसमें लिया है tough word, but it is going to be tight and tough, but it is loose and thin. क्यों? अगर दोनों पेप्टिडोग्लाइकन के होते तो दोनों का नेचर एक होता, बट नहीं है. Because along with peptidoglycan, capsule is something which is made up of two with lipopolysaccharide. That is very, very, very important. That's the composition of capsule. Glycocalyx capsule is being made up of lipopolysaccharide along with peptidoglycan. But slime layer is something which is only made up of peptidoglycan, and that's the reason it is going to be loose and thin. So that's one thing that is important, right? And then उसके बाद तेरा glycocalyx and all these again are similar to fimbria. This is something that help in colony formation. ठीक है? It helps in colony formation, right? You can there are different types. Lipopolysaccharide, या फिर there is sometimes we write it as polypeptides, so it's not going to differ. Okay, polypeptides different, बस वही है, उसका एक extra composition है along with peptidoglycan. It is made up of this, right? Okay, next is cell wall. देखो यार, it's going to be important. The cell wall, very, very, very important. The cell wall is going to be rigid in case of bacteria. It is going to be made up of peptidoglycan, that is going to be murin, right? हमेशा देखना, I just told you, just now I told you, the gram positive bacteria, the cell wall is not going to be well developed. On the basis of gram stain, we have two types of bacteria. I'll be discussing it later on in a bit detail. हमेशा रखना, that gram positive bacteria is going to have only single layered cell wall. It is going to have single layered cell wall, whereas the gram negative bacteria is going to have double layered cell wall. NCERT exemplar is going to be very important. Okay, please just remember this. The gram positive have only single layered cell wall. Gram negative have the double layered cell wall. This double layered cell wall very, very, very important. अब double layer का मतलब एक होगा inner layer और एक होगा outer layer, right? तो हमेशा 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 याद रखोगे, the inner layer is going to be something which is made up of the inner layer is going to be made up of peptidoglycan, and the outer layer is going to be made up of lipopolysaccharide. It's very important, very, very, very important that you have to understand, right? The gram positive, gram negative bacteria have inner and outer layer. The inner layer is going to be made up of peptidoglycan, outer layer is going to be made up of lipopolysaccharide. ठीक है? अब होता क्या है ना, अगर कभी भी कोई एक क्वेश्चन आया था, एक ये भी क्वेश्चन बहुत अच्छा क्वेश्चन था, AIIMS 2015 में था, I don't remember exactly. The cell wall, see, अगर तू cell wall से if you take a bacteria, by the action of an enzyme, you can just destroy its cell wall. मतलब the bacteria will be having no cell wall. This particular form of bacteria, which is having no cell wall, the bacteria without cell wall, is called as L form bacteria. It's again a question आया हुआ है, so that's why I'm giving it right over here. The without cell wall is going to be called as L form bacteria, and the enzyme used for it is lysozyme. So ये important है, okay? So you should remember, it's even given in NCERT practicals. So it's better if you remember it. Okay, L form matlab lister form. Okay, lister form bacteria, L form bacteria. So it's important. Okay, so this is one thing. Then cell glycocalyx हो गया, cell wall हो गया. Let's discuss cell membrane. That is again important. The cell membrane की अगर मैं बात करूँ तो cell membrane मैं बता चुका हूँ, it is going to be analogous to human lipoprotein. So बस उसमें और कुछ नहीं है. The cell membrane का अंदर का part, that is going to be cytoplasm. It is important. I discussed right over there. Cytoplasm is having always no membrane bound organelles like mitochondria, ER, lysosomes, Golgi bodies, all will be absent in case of bacteria. क्या होते हैं उसमें, that is important. एक चीज हमेशा याद रखना, there is something called as streaming or cyclosis. I'll be discussing in cell bio chapter. That is going to be absent in case of bacteria. The streaming or cyclosis is going to be a characteristic property of only eukaryotes, not of prokaryotes. Okay, so that is going to be absent in case of bacteria. So the very first part is very important in case of cytoplasm is going to be mesosomes, right? अगर मैं mesosomes की बात करूँ बेटा देख, it's going to be very simple. Mesosomes are called as the functional mitochondria. The functional mitochondria, because just now I told you cytoplasm have no membrane bound organelles. Mitochondria is going to be a membrane bound organelle, right? But that is going to be absent. But the bacteria lives. So how does it live? Because it is having something called as mesosomes. Mesosomes take place of mitochondria in case of bacteria. It is the functional mitochondria. AIIMS assertion and reasoning. Assertion saying that mesosomes act as the functional mitochondria. Reason for this is going to be very important, because the mesosomes have oxidative enzymes. The mesosomes have the oxidative enzymes for respiration, and that's the reason why it acts as functional mitochondria. On the basis of positions, the mesosomes are in two types. One is called the peripheral mesosomes. One is called as the peripheral mesosomes. Another is something which is called as the central mesosomes. हमेशा the peripheral mesosomes are going to be, near the cell membrane, whereas the central mesosome will be something which is deep buried inside the cytoplasm. Deep buried inside the cytoplasm. So can I say one thing? अगर वो deep buried है, if it is not easily available, मतलब वो कुछ तो काम ऐसा करेगा जो chromosomes से related होगा, right? That is polyamines, not true chromosomes. So हमेशा याद रखना, central mesosomes is something that is used in DNA replication and cell division. The DNA replication and cell division is the function of the central mesosomes. The peripheral mesosomes functions to be as, its functions for peripheral mesosomes functions for cell wall secretion and cell respiration. Cell wall secretion and cell respiration. ये direct NCERT examples I'm just giving it right away. It's going to be very important for you, right? So एक चीज और है जो तुम्हें याद रखना है बच्चे. देख, अगर this is the bacteria, right? अगर मैं मान रहा हूँ कि उसका cell wall है, ठीक है? Inside is going to be your cell membrane. हमेशा याद रखना, the cell membrane will invaginate like this. The cell membrane is going to invaginate like this. This invagination of cell membrane is something which is called as mesosomes. समझ आ रही है बात? मतलब क्या है? Mesosomes is something that secrete the cell wall. And इस mesosomes को कौन secrete करता है? Cell membrane forms mesosomes in form of, that is again very important, tubules, vesicles and lamellae. NCERT liner, NCERT exemplar. So mesosomes are something which is secreted by cell membrane in tubules, vesicles and lamella, similar to Golgi bodies. So can I say one thing? The major part of this particular mitochondria and Golgi body is something which is just handled by mesosomes. So mitochondria and Golgi body is something, in case of eukaryotes, is similar to mesosomes in case of prokaryote. The cell wall secretion and cell respiration में कौन help करता है? Is going to be mesosomes. Which mesosomes? Peripheral mesosomes. That's going to be important, right? So I guess that's it with mesosomes. The second part that you have to remember is going to be the storage granules. Okay, storage granules. NCERT for biological classification chapter has used storage granules for. The same NCERT in cell bio chapter he used inclusion bodies term. So that is going to be important. हमेशा रखना, the inclusion body terms is something that is used for storage of reserved food material. It's going to be very important. For the storage of reserved food material, we use something which is called as this one, right? Inclusion body. अब inclusion body on the type of food which is stored, it is going to be two types. Okay, inclusion bodies are going to be two types, right? So one is called as your glycogen granules. Another is something which is called as, this one, volutin granules. Glycogen is going to be volutin granules, right? The glycogen is something which is a carbohydrate, so it is going to store carbohydrate, right? And volutin is something, volutin is something we call it as metachromic pigment. It's going to be very important. Why? Because it is going to be colored, and colored मतलब हमेशा देखना volutin metachroma pigment is going to be phosphate. It is going to store phosphate. It's important, right? It is going to store phosphate. So that's one thing. Then उसके बाद अगर cytoplasm में, then it is going to have nucleus. I told you nucleoid, no true chromosome is present, right? I've already told you in general characteristic, incipient nucleus, genophore, है ना, naked nucleus, and मैंने क्या बताया अभी बताया था, there is no true chromosome. Instead of that, false chromosomes are present along with polyamines, that is non-histone protein, and the type of DNA which is called as DS circular DNA, naked DNA. हमेशा एक चीज तुम्हें याद रखनी है. See, because of this, देखो यार, important है. See, because there is no particular the bacteria, like जो bacteria है ना तेरा, in this particular type of bacteria, there is no true this one. In case of bacteria, there are no true chromosomes present, right? Since there are no true chromosomes present, there is polyamines present. हमेशा याद रखना, there is no true chromosome present. There is no membrane which is present around the nucleus. That's why they show a type of curve. A growth curve, very 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 important. A type of growth curve which is being shown by the bacteria is going to be very 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 suddenly it is going to increase, right? Then it is going to be stationary, it is going to be normal, it is neither increasing nor decreasing. Then it is going to decline, stationary and decline. So the growth curve, अगर मैं एक तरह से लूँ तो decline से पहले तक कर लें और उसको थोड़ा सा अच्छे से बनाएं. So we are going to get this type of curve. We are going to get this particular type of curve. And this type of curve is called as sigmoid curve. So the growth curve which is being shown by the bacteria is going to be a sigmoid curve. The very first step is going to be log, which is going to be log, stationary, and then your decline phase. This is going to be the, ये arrange करने को आया है question बेटा, I guess 2017 में ही आया है KCET में. So I'm just telling you this, it's going to be very important. Log, stationary and decline. The type of curve is being shown by eubacteria is going to be sigmoid curve. Why? Because there is no true chromosomes which are present. Instead, polyamines are being present right over there. So it's going to be important for you to understand, right? Then if you wish, this is important. उसके बाद तो बेटा कुछ नहीं है. उसके बाद तुम्हें कुछ concepts याद रखना. That is going to be plasmids. तो मैंने ये बता रखा है anyway. I'll be teaching this and all in this one biotech a bit detail. Just have a glance. Plasmids were something which were discovered by Lederberg couples. पति पत्नी दोनों, Lederberg. Okay. So plasmids were discovered by them. हमेशा देखना plasmids are those which have self-replication ability. They have self-replication ability. Okay. Another thing that is important right over here is going to be your type of DNA which is present. Plasmid have again the same DNA, DS circular, naked DNA, right? So it is having a self-replicating ability. ये important है. एक और चीज जो बहुत important है बेटा देख. There are two types of bacteria. Okay, consider there are two types of bacteria, right? One is that particular type of bacteria, which is having this particular on the basis of the fertility ability, and on the basis of phenotypic character. Bacteria are classified in two types. One is that particular bacteria which is present with F factor. One is that particular bacteria which is present with F factor. Another is that particular bacteria which is absent with F factor. F factor is absent in case of one bacteria, F factor is present in case of one bacteria. The bacteria in which F factor is present is called F plus bacteria, is going to be called as F plus. One जिसमें F factor is not present is going to be F negative bacteria, right? अब consider there is DNA in case of this bacteria. अब इस DNA के साथ मैंने लगा दिया एक desired plasmid. Along with this DNA, I want a character in the bacteria. I use it of my choice. अब क्या होता है? By a particular process of conjugation tube, right? There grows a tube from F plus to F negative bacteria, and what happens is this particular DNA now moves to F negative bacteria, where it fertilizes the ova and the bacteria divides further, right? Kind of. I'm just giving you the analogy, fertilize wala ova and all. There's no ova and all in case of bacteria. I'm just giving you the analogy right over there, that this particular F plus bacteria moves into this particular F negative, and it is going to later on divide. From matlab F positive is going to, jab F positive enters the DNA content of F positive and moves to F negative, then only the bacterial division or reproduction get completed. अब क्या होता है देख, now this particular has moved and ये bacteria divide कर गया, right? The process is complete. The conjugation tube will degenerate. इस bacteria में क्या बचा? अब F plus नहीं रहेगा, right? The particular type of bacteria which was called as F plus after completion of the conjugation technique will be now called as HFR cell. It's going to be very important, very important. ठीक है? It's going to be seriously very important. F plasmid, ठीक है? The HFR cell, the particular type of bacteria. और मैंने बताया था यहाँ पे जो मैंने plasmid use किया था, right? Along with the bacteria. When bacteria के DNA के साथ मैंने एक plasmid लगाया था, इस particular type of cell को अपन बोलते हैं episome. ठीक है? ये सब आए हुए question हैं, so I'm just giving you right over here. It's bacteria से related है, question पूछ सकता है, right? And present with F factor is going to be HFR cell. The particular bacteria which F plus bacteria after conjugation is called HFR cell. Plasmid अगर DNA के साथ लगा दिया तो episome, और फिर जब F plus का DNA कट के F minus में जाता है, it is going to divide and going to reproduce, right? So it's going to be important right over here. ये important है. There's one thing, okay? So now let's directly go to our very important topic, which is called as gram staining. Okay, so gram staining. देखो यार, कुछ नहीं, it's going to be very simple. ठीक है? The gram staining. Yeah, so gram staining, right? So it's going to be very important. This is one of the very favorite topic for any of the examiner to ask. So you should know this, okay? Remember, H.C. Gram was the person who discovered this particular staining. That is why it's called as gram staining. The very important direct NCERT practicals, the steps of gram staining. Very first step is to use a particular stain which is called as crystal violet. Right? It's going to be very important, crystal violet or gelatin violet. Very important. Prepare on the bacteria slide. Okay, prepare सबसे पहले एक bacteria slide लो, उस पे डालो crystal violet और gelatin violet. Very first step. Second step, pour the solution of potassium iodine. It's going to be again very important. Take सबसे पहले crystal violet और gelatin violet. Then in the second step, pour potassium iodine solution. The very third step that is going to be there is going to wash it with acetone and then ethyl alcohol. It's going to be very important. Acetone are going to be ethyl alcohol. The fourth step, there will be a bacteria. The bacteria, right? There will be two categories of bacteria that we will get, right? I'll discuss it right over here, everything, so it will be easy for you. Bacteria, I'll get two particular types of bacteria. एक stain वैसा होगा, bacteria which will retain the stain. Okay, the bacteria that retain the stain, There will be one bacteria that will not retain the stain. Retain the stain मतलब after this completion of the staining, the bacteria which is going to still show positive, मतलब gelatin violet or crystal violet color ही दिखाएगा, that will be called as gram positive bacteria. But one which do not retain the stain will be called as gram नेगेटिव बैक्टीरिया. It's going to be very important, right? So मैंने तुम्हें इन दोनों के बीच के में सारे पॉइंट्स दे देता हूँ, right? ये रिटर्न करता है नहीं करता. उसके बाद मैंने पहले बताया था ग्राम पॉजिटिव इज समथिंग which is having single layered cell wall, right? It is going to have single layered cell wall, and the gram negative is something which is going to have double layered cell wall. Then उसके बाद मैंने बताया था कि सेल वॉल इज समथिंग व्हिच इज डबल लेयर्ड, द इनर लेयर एंड आउटर लेयर. द इनर इज समथिंग व्हिच इज मेड अप ऑफ पेप्टिडोग्लाइकन एंड द आउटर इज समथिंग व्हिच इज मेड अप ऑफ लिपोपॉलीसैकेराइड. तो हमेशा याद रखना द लिपिड कंटेंट इज गोइंग टू बी हाई, द लिपिड कंटेंट इज गोइंग टू बी हाई इन केस ऑफ this particular type of bacteria, but the lipid content is going to be low. The lipid content is low, instead peptidoglycan का content is going to be high in case of gram positive bacteria. That is one point. उसके बाद तुम्हें अगर याद रखना है तो उसके बाद the thickness, the cell wall thickness, the thickness of the cell wall, ठीक है, the thickness of cell wall is going to be around 20 to 80 nanometer. Whereas the cell wall thickness over here will be less, 8 to 12 nanometer thick, right? उसके बाद अगर तुम्हें याद रखना है तो there is something lipid content यहाँ पे कम है, यहाँ पे lipid content is high, मतलब 70 to 80% is going to be lipid, but यहाँ पे 70 to 80% is going to be glycogen. Then उसके बाद एक बहुत ही important question, टेक्नोइक एसिड, ठीक है, बस टेकोइक एसिड, very important, is going to be present in cell wall. The cell wall is something which will be having teichoic acid, but the teichoic acid is going to be absent in cell wall in case of gram negative bacteria. So ये एक चीज है जो तुम्हें याद रखनी है. उसके बाद तो बेटा कुछ है, उसके बाद मैंने पहले बताया था gram positive is something which is present only with inner rings, right? S and M rings are only present, but here L, P, S, M rings all are going to be present in case of gram positive bacteria. So these are something which are this one. उसके बाद हमेशा के हमेशा याद रखना all pathogenic bacteria, all नहीं, most word will be more better. NCERT exemplar directly from biotech chapter. Most of the pathogenic bacteria belong to gram negative group. Less pathogenic bacteria belong to gram positive group. Yeah, हमेशा important point है, that is going to be. So these are all the points which are important and direct. Only इतना ही NCERT practical have given और मैंने वही points तुम्हें यहाँ बताए हैं. So बस इतना ही पढ़ना, कोई भी बाहर के questions मत पढ़ना, okay? बस इतने ही से question आएगा और कहीं से नहीं आएगा, okay? Because it's only given in NCERT, right? So that's one thing that is going to be your gram staining, okay? So we are done with gram staining. Next is going to be the nutrition for bacteria. That is important, right? So देखो यार, answer is going to be very simple. The gram staining bacteria, देखो यार, the nutrition, it's very important. The nutrition, अगर मैं बैक्टीरिया के न्यूट्रिशन की बात करूं, हमेशा रखना the bacteria shows something called as metabolic diversity. The metabolic diversity is being shown by the bacteria in case of nutrition, but there is something which is shown by protista, which is diversity in something else. I'll be discussing it. Fungi का diversity किसी और में होता है, तो I'll be discussing it later on. हमेशा रखना nutrition में bacteria diversity दिखाता है. It's going to be very important. 90% of bacteria are going to be heterotrophic. Very important. Most of the bacteria show heterotrophic mode of nutrition. There are autotrophs for sure, but most of them are going to be heterotrophic. Okay, so that's one thing that is important, right? Okay. Now let's directly discuss all the types of. सबसे पहले है तेरा autotrophic mode of nutrition, right? Autotrophic mode of nutrition. These are of two types. One are called photoautotrophs. One are called photoautotrophs. Another are called chemosynthetic autotrophs. Very important. ये तुम्हें समझना है. Chemosynthetic autotrophs, right? Photoautotrophs, it's going to be very important. Photo means they are going to use light energy. They use light energy for synthesis of food. Chemo, they are going to use chemical energy. They are going to use chemical energy for synthesis of food. हमेशा याद रखना है, ये बहुत important point है. They are going to use light energy. These are going to use chemical energy, right? अब since these are using light energy, can I say one thing? They are performing a kind of photosynthesis which is similar to plant, right? But this is wrong, because तो प्लांट फोटोसिंथेसिस जो करता है ना, इट इज गोइंग टू बी ऑक्सीजेनिक फोटोसिंथेसिस, बट द बैक्टीरिया फोटोसिंथेसिस इज गोइंग टू बी non-oxygenic photosynthesis. NCERT liner, NCERT practical, NCERT exemplar is going to be very important. The bacteria perform a type of photosynthesis which is going to be non-oxygenic photosynthesis, whereas the type of photosynthesis which is being done by your plants is going to be oxygenic photosynthesis. So it's going to be important point. Okay, and देखिए ध्यान से the type of होता क्या है ना, why we say this, since it is doing non-oxygenic photosynthesis, I've given you, I have to prove you this. So listen, it's being class 11th plant physio, I'll be teaching right over there. Photosynthesis करके एक chapter है, उसमें many people have proved this. The particular type of oxygen which is released during photosynthesis comes from water, not from the glucose or the carbohydrate which we use during the sugar is something which is converted to CO2, H2 वगैरह, oxygen. तो हमेशा याद रखना, it is the particular type of water from which we get oxygen. If the source of this particular composition of water changes, consider if it is going to be H2S, then there will be no oxygen which will be produced. So यही reason है photoautotrophs uses H2S for synthesis. They use light energy, uses H2S as an inorganic compound as well as some organic compound and produce output. This one is H2S is going to be converted by using light energy into food, and that's the reason why we are saying that it is going to perform non-oxygenic photosynthesis. Okay, the chemical energy, chemosynthetic autotrophs, what they do is, देखो ये point बहुत important है बेटा. The chemosynthetic autotrophs, what they do is they use the chemical energy, and the term is going to be very important, very important. They oxidize the chemical compounds. NCERT exemplar, chemosynthetic autotrophs performs oxidation. They oxidize the chemical compounds and obtain the food by converting chemical energy into their food. Right? So it's going to be some chemical compounds are converted into their own food by So now let's move on to reproduction, right? So reproduction, as in case of bacteria, is going to be very simple. The reproduction, two types, asexual and sexual reproduction. So let's first discuss on all the major asexual reproduction techniques, right? Asexual, कभी भी है, there are two major techniques. सबसे पहले binary fission is going to be very important. अगर binary fission, it's the most common technique, which is being used by all the bacteria worldwide. सबसे ज़्यादा use करने वाली technique by all the bacteria is going to be binary fission. हमेशा रखना binary fission होता कैसे? If there is one bacteria like this, right? This bacteria is going to divide by a particular process of central septum. That is very important. सबसे पहले ना एक septum बनाता है ये bacteria, and from this septum, okay, there is a particular type of septum which is being formed, that is called as transverse septum. The transverse septum is being something which is being formed by the bacteria during binary fission. This transverse septum grows from central towards the outer side, and this bacteria will then later on divide along this particular type of septum, right? So when I ऐसे ले रहा हूँ ये line, then there will be, this is called binary fission, right? This particular type of is called binary fission. समझ रहे क्या बात? That is your binary fission. Remember, this is something which is in favorable condition by binary fission. Again, in favorable condition, another type of condition, the cell bacteria again sometimes divide the asexual mode of reproduction. There is a particular type of process which is called as amitosis. NCERT exemplar. Amitosis is a technique of division of genetic material. The division of genetic material, and this division of genetic material is going to be unequal. NCERT exemplar, amitosis is the unequal division of the genetic material. हमेशा याद रखना, and amitosis is going to be faster than mitosis as well as meiosis. Mitosis and meiosis. It's important right over here. ठीक है, तो तुम्हें समझना होगा mitosis, meiosis. I'll be discussing in cell biology chapter, but you have to remember it is always faster than mitosis and meiosis. Mitosis is division of the genetic material, unequal division of the genetic material. That is one thing. Then third is ये दोनों favorable condition में. अब आते हैं unfavorable condition में. There is something which is called as endospore formation. there is something which is called as endospore. तो हमेशा के हमेशा बेटा याद रखोगे, this is something binary fission, यहाँ से लेके यहाँ तक, it is going to be in favorable condition. These are something which is in favorable condition. This is something which is formed in unfavorable conditions, right? It is something which is formed in unfavorable conditions. So it is going to be very important. देखो it's simple a bit, right? तुम्हें थोड़ा समझना हो. So देखो this one diagram is important. For endospore, this diagram is going to be really very important because this was directly asked in AIIMS 2003. So I'm just giving it right over here to you. Again. So ये diagram NCERT में नहीं दिया, but it's given very nicely in NCERT practical, so it's important. Remember, this is something which is called as core. This is called as core. ठीक है? The core is something, core is the main part, which is something which is similar to cytoplasm, right? The covering of this core over here, that is called as core membrane, right? The right over here, the blue part, this is called as the core wall or core cell wall kind of, right? उसके बाद आता है तेरा ये the inner core coat, the inner core coat, then because the outer core coat, the outer core coat, the red वाला is going to be very 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 important. हमेशा रखना the most toughest layer, exosporium basal layer, exosporium basal layer. ठीक है? तो in unfavorable condition the bacteria is going to form this. यहाँ पे है तेरा bacteria का cytoplasm, right? These when you layer in unfavorable condition, it is very tough to break this and all off. So only in favorable condition the bacteria, they come out of this and they can survive within this for many years. हमेशा याद रखना, there is something which is very important, right? I have to tell you. Between, so there is something which is right over here. It's going to be very 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 important between the core wall and inner cell wall. This green part, ये बहुत important है बेटा. ये question आया हुआ है. I guess PMT 2015. This was a direct question. देखो ध्यान से, ये green वाला part is something which is called as cortex. This cortex is having बहुत 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 important है. The cortex during endospore formation is made up of calcium diploconate or calcium diploconalic acid, which gives it the very particular type of tough protection which protects the particular bacteria in unfavorable condition. तो एंडोस्पोर पढ़ाने का यही एक रीजन था कि ये पर्टिकुलर ग्रीन एरिया बताना, बिकॉज कॉर्टेक्स क्वेश्चन आया है. तो द कॉर्टेक्स विच इज प्रेजेंट, ये मार्क करने को भी आ सकता है. द कॉर्टेक्स इज समथिंग विच इज प्रेजेंट बिटवीन द core wall and inner core coat, and it is going to be something which is made up of calcium diploconate or calcium diploconalic acid, right, which gives it the protection in unfavorable conditions. So these are the types of asexual mode. In the best is endospore formation, very protective, and the most used is by binary fission, right, most common. So these are the type of asexual reproduction. So like you can make it off if you want, or you can wish, right? Okay. So now let's move on to the sexual mode of reproduction. मैं वो sexual में मुझे पता है तुम लोग क्या सोच रहे होगे. It's like the three ways, Griffin's experiment and all मैं वहीं पढ़ाऊंगा, तो यहाँ पे पढ़ाने वाली कोई बात है नहीं. ठीक है, sexual mode में देख, सबसे important is going to be sexual mode of reproduction. हमेशा देखना, the sexual mode of reproduction is actually absent in case of bacteria. Instead we call it as by a name of something called as genetic recombination. हमेशा रखना, the sexual mode of reproduction is absent. Instead we use a term for that is called as genetic recombination. This occurs by three ways. One is called as transformation. One is called as transformation. The other is called as transduction. Transformation, transduction, and third is something which is called as conjugation. I guess I should. So transformation, remember the transformation is a particular type of process where the DNA of dead bacteria, बहुत 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 important है बेटा, DNA of dead bacterial cell is something which is incorporated to DNA of living bacterial cell. That process, I guess, definition is enough for this, right? DNA of dead bacterial cell is something which is incorporated into DNA of living bacterial cell. This particular type of process is called as transformation, and we will discuss in a bit more detail about this in molecular bio chapter, class 12, right? So genetics, so transformation was something which was proved by Griffin's experiment, so we'll be discussing it. Transduction is out of syllabus, so as per me, no need to even study it. ठीक है, इस पे बिल्कुल मत पढ़ो. On conjugation है, तो मैं अभी just पढ़ाया था HFR cell and all, right? HFR cell, episomes वही है, इसमें और वही उसी को कहते हैं conjugation. हाँ यार, तो conjugation is something which is HFR cell and episomes, right? उसके बाद कुछ नहीं है. That's it with this video. इसके आगे it's okay. Right, there are two more topics which are remaining, which is BGA and mycoplasma. I'll be discussing it in my next video, mycoplasma and BGA, because these are very important. और archibacteria, ये तीन है that is going to be very important. तो वो मैं अलग से discuss करूँगा. And economic importance of bacteria तो मैं उसी के साथ discuss करूँगा. Right, so yeah, that's major part I have discussed right over here for bacteria. So 30 years question पूरे हो गए मेरे इसके. So I guess try out solving most of the question. The question link will be there in the description for Kingdom Monerans. Try out solving them out. Right? They are good questions, good quality questions, and the answers for those particular questions will be available on my next video. So again, वही आपको रहने वाली बात. Hope you like the video. Like, share and subscribe as per your wish. Right? अच्छा लगे तो बताओ कमेंट में, ना भी अच्छा लगे तो भी बताओ कमेंट में. कोई कुछ नहीं